Hey guys, so we're going to do the second part of photosynthesis, and we're going to talk specifically about how sunlight is converted into glucose or other sugars that can be used by the plant. Just a basic overview of what photosynthesis is. Remember, that's the process of taking in light, carbon dioxide, and water to produce sugars. Most of the time that's going to be a glucose molecule, but it could be sucrose as well. As a byproduct of photosynthesis, we're going to produce oxygen. And so this diagram just kind of simplifies the overall process. We have two main parts of photosynthesis. You have your light-dependent reactions, which are going to be the reactions that occur and directly use the light. And then we have these light-independent reactions, which are going to be the reactions that occur that do not directly use the light energy that was originally absorbed. So let's break down the light-dependent reactions. These reactions are going to occur in the thylakoid membrane of plant cells. And within the thylakoid membrane, you have these light harvesting complexes. And a bunch of these light harvesting complexes are going to have chlorophyll molecules attached to them. That could be either chlorophyll A or chlorophyll B, or maybe carotenoids or any accessory pigments. A bunch of light harvesting complexes together with their chlorophylls attached is going to give you a photosystem. And there are typically two photosystems that we talk about in these types of reactions. So if you remember, light can travel in pockets of light called photons. So those photons are going to be absorbed by chlorophyll A and chlorophyll B. Well, these types of chlorophyll, when they absorb that photon, they move from a ground state to an excited state. And then they immediately want to move right back down to their ground state. And that moving back down is going to release energy. And they do this really quickly. So eventually, these molecules are going to release energy back and forth. And it's going to bounce around to different chlorophyll molecules. And eventually, it's going to hit the reaction center, or the chlorophyll reaction center, which we denote as CHL. Once that CHL is going to become excited, we annotate that with the CHL with an asterisk symbol above it. So that chlorophyll in the reaction center actually gets oxidized. So remember, that's when it's going to lose an electron. As it gets excited, it wants to hand that electron over to another electron acceptor or electron carrier. We talked about an example as NADH. So the chlorophyll in the reaction center becomes oxidized or loses an electron, and the carrier is going to pick up that electron or become reduced. A little bit more detail about those photosystems that we were talking about. So the photosystems make up the light-dependent reactions. There are two photosystems. The first photosystem in which light interacts with is photosystem 2. And this is going to use chlorophyll known as P700. And what that means is that this type of chlorophyll is going to absorb the wavelength of light at 700 nanometers. And it's going to use a process of non-cyclic electron transport. And we're going to talk about what exactly that means in the next slide. Photosystem 1 is the second part of the light-dependent reactions, and this is going to be made up of a P680 chlorophyll. And what that means is that light's going to be absorbed at 680 nanometers in this type of chlorophyll. And this is the photosystem that is linked chemically and spatially to photosystem 2. And we're going to talk about how that occurs in the next diagram. So here's a general overview of what these two photosystems look like in the thylakoid membrane. So light is going to travel into the thylakoid membrane through a photon. That photon is going to interact with the chlorophyll molecules. It'll get bounced around between all of the different chlorophyll molecules until eventually it hits that reaction center. So once it hits that reaction center, the reaction molecule, or the chlorophyll, becomes excited. And when it becomes excited, it's going to hand over that electron or lose an electron, and that's becoming oxidized. This electron carrier is going to become reduced, and it's going to move that electron down a series of steps known as an electron transport chain. You might recall that from biology, but we're going to talk more about that when we talk about cellular respiration. In this process, water is used, or water is split by sunlight, and that is actually where oxygen is given off. So oxygen is given off as a waste product, and it's not intended. It's not an intended product of this reaction. So as that molecule moves down the electron transport chain, it's then going to get passed on to the P700 chlorophyll. This chlorophyll reaction center here is going to absorb light at 700 nanometers, and it will then also become excited. And when it becomes excited, it's going to hand that electron over and become oxidized. That electron carrier is going to move that energy through another cycle known as the Calvin cycle, which is what we'll talk about. And in that process, we're producing NADPH. This process here is known as non-cyclic electron transport because that's just passing an electron to get through to photosystem 1. Well, photosystem 1 can actually send that energy that the chlorophyll's reaction center was trying to give off, it can actually recycle that to produce a lot of ATP through a process known as chemiosmosis. Again, we'll talk about that with cellular respiration, but the important thing to remember is that this cycle here can be cyclic, meaning the reaction center chlorophyll can hand over that electron to an electron acceptor, which can then come back around to create ATP through a gradient. And that's important because the next cycle requires a ton of ATP. So this here, being cyclic, allows us to recycle or keep making a lot of ATP. The second part of this that we're talking about are the light-independent reactions, and specifically this is going to occur in the stroma, also known as the Calvin cycle. And like all cycles, they're going to be regulated by enzymes, and there are a couple of main steps that occur in the Calvin cycle. The first is that carbon dioxide is going to be accepted by a molecule, RuBP, and that reaction is going to be catalyzed or sped up through the enzyme known as Rubisco. Rubisco is actually really interesting. It makes up most of the matter in leaves of plant cells where photosynthesis occurs. It's also the most abundant protein in the world. This catalyzed reaction here is going to then produce a molecule known as a 3PG molecule. We'll talk about all of these names in the diagram in just a minute. That 3PG is going to be reduced, so it's going to gain an electron and become a G3P using a lot of ATP through that cyclic formation of ATP in photosystem 1, and also some NADPH, which is an electron carrier similar to NAD or NADH. That G3P that was produced is then going to be used to produce all of the sugars the plant needs, and that's going to be about 1/6 of all the G3P. The other 5/6 of the G3P is going to be used to create RuMP, which is going to then be phosphorylated to RuBP, which can then accept more carbon dioxide. So it's a way of reusing some of the materials that are being produced. So for this last slide, we're going to talk about the Calvin cycle in a little bit more detail. Just to remind you, carbon is going to enter at a given time, and it's going to be accepted by RuBP, and that reaction is catalyzed by Rubisco. That is going to then form 3PG, which stands for 3-phosphoglycerate. That 3-phosphoglycerate is going to be broken down in 2 steps using ATP and NADPH to form G3P, which is glyceraldehyde 3-phosphate. That G3P is going to be used to produce sugars, and some of that, again, remember, is going to go back around to form RuMP, which can then be phosphorylated or add a phosphate to form RuBP. So here's just the general reaction of the Calvin cycle that occurs in the stroma. Make sure that you guys are reading along in your book. A lot of new vocabulary, a lot of terms that you guys need to be familiar with. And your book also has really good diagrams if these diagrams did not.